Next up, Zeineb is actually back. So we will have Zeineb from Abdelmalek Essadi University in Morocco, presenting on Th1, Th17 imbalance in systematic lupus, erythematosus, and lupus nephritis. Zeineb, feel free to share a screen and take away.
Hello, everybody. I'm Professor Zeineb Zian from the Faculty of Sciences and Techniques of Tangier at Malik Saud University in Morocco. And first of all, I want to thank Bion Sciences Initiative for giving me this opportunity to present my work about Th1, Th17 imbalance in systemic lupus erythematosus and lupus nephritis. Okay. Okay. So systemic lupus erythematosus, or simply lupus, is a complex autoimmune disease that involves multiple organs, and anti-nuclear antibodies are the key cause of this disease, where the body's immune system attacks its tissue. Scientific evidence has shown that anyone can have or develop the lupus, but it affects women more often than men. So in fact, 9 in 10 people with lupus are women, and the mean age is 30 years. And the disease is typically diagnosed between 15 and 45 years of age. So the prevalence of the disease is 2 to 3-fold higher in Blacks and Hispanics than whites. So overall, there is a strong genetic predisposition of the lupus. There are specific alleles, such as HLA, which has been reported to be highly associated with the onset of the lupus, and also different severe environmental factors like infections, drugs, or exposure to sunlight. So inflammation and autoantibodies can cause damage to any organ, but most often affect the skin, the lungs, kidney, blood cells, and also brain. So clinical manifestations of lupus is highly variable and sometimes very dependent on the body system that it's affected, and many may include fatigue, fever, hematologic manifestation also, like this. For example, hematologic manifestations or symptoms are present in 100% of patients. Arthritis is present in 80% of patients, and also the renal complication, which is present in 60 to 70% of patients. And when we talk about renal complication, we talk about lupus nephritis. So what is lupus? Lupus nephritis is a severe complication of systemic lupus erythematosus caused by the deposition of immune complexes in the kidney, in the glomerulus, and mainly the immune complexes can be anti-nuclear antibodies and anti-double-stranded DNA, complement activation, and inflammatory cells. So in general, lupus nephritis occurs in 50% of patients with systemic lupus erythematosus, and more than half of patients with lupus develop lupus nephritis within 10 years of lupus disease, and more than 75% will over their lifetime. So 4 to 10% of lupus nephritis patients will develop end-stage renal disease, and the incidence is higher in Blacks, Asians, and in Hispanics. So in general, systemic lupus erythematosus with or without renal involvement is characterized by increased fraction of circulating Th17 cells and elevated levels of IL-17A in lupus patients, and correlation between Th17 cells levels and disease activity. Also is characterized by significantly increased frequency of CCR6 positive and CXCR3 positive in patients with lupus and with DNA positive, and also a positive correlation between ROR-gamma T, the transcription factor, the expression of this factor and the IL-23R receptor. So that's the aim of this work, to investigate the TH cell subpopulations, I mean TH1, TH2, and TH17 cells in Tunisian patients with and without renal involvement, and also investigate their functional association with kidney damage through the expression of their specific markers, evaluating also the mRNA expression of ROR-gamma T and IL-23R. So to do this, we recruited 9 patients with active lupus during 3 months from the internal medicine and nephrology departments. And for the lupus patients, so all the patients satisfied the classification criteria of American College of Rheumatology. And for the lupus nephritis, it was confirmed by renal biopsy and the classification of lupus by the International Society of Nephrology Renal Pathology. Also included healthy controls, and for the diagnosis of patients, it was based on the clinical presentation are results of some analysis to the anti-nuclear antibodies and anti-double-strand DNA. Then subjects were classified or subdivided in 3 groups. So we had 5 healthy controls and 9 patients, including 4 with renal involvement and 5 without. Then we did the isolation of PBMC, so peripheral blood mononuclear cells were isolated from blood, and then were separated, stimulated, differentiated, and then incubated with or without stimulation. After that, regarding the TH17 immune phenotype cells, the PBMC were signed by a combination of specific antibodies, of surface markers and intracellular cytokines, and then subsequently analyzed by the flow cytometry. So for the QRT-PCR, after isolation of the PBMC, the total RNA was extracted for the PBMC, and then we did the reverse transcription, and then the QRT-PCR was performed in a final reaction volume of 20 microliter. So here we had the main demographic and clinical characteristics of our patients. So as you can see here, the main age of the onset of the disease was 9 years. The malar rash was observed in half of patients, and all the patients had the ANA, anti-nuclear antibody positive, and the high level of the positive anti-double-stranded DNA was detected in more than half, and we had 4 patients that presented the renal involvement. This is the results of the flow cytometry, and as you can see here, the TH17 cells were level in lupus with and without renal involvement was elevated and higher than healthy controls. Also for the TH1 cells was higher and significantly higher in lupus patients with or without renal involvement than in healthy control. Here we can see this difference clearly, and we can see also that the TH2 cell frequency showed no difference between the 3 groups. I mean the lupus, the 2 subgroups of lupus, and the healthy controls. So here we have the TH subsets in simulated PBMC of lupus, the 2 subgroups of lupus and healthy control, according to the expression of chemokine receptor CCR6 and CXCR3. So the proportion of CCR sex positive and CXCR3 positive T cells were significantly higher in lupus patients compared to healthy control. And here we have the results of three subsets of cells. So we have here the TH1, TH17, and TH117. So the TH117 has the phenotype of CCR sex positive and CXCR3 positive. And as you can see, these we have the different TH subsets that indicate that TH1, TH17 cells were significantly increased in lupus patients than healthy control. But also the TH17 is only significantly increased in lupus patients compared to healthy control. But there is no significant difference between the patients group, the patients group, I mean the lupus nephritis and lupus patients. For the relative mRNA expression of ROR-C and IL-23, so we observe and we found that the expression of ROR-C and IL-23 are elevated in lupus patients compared to healthy control, but there is no significant differences in these parameters between the two groups, I mean the lupus patients and the healthy controls. So at the end, we can see that although the lupus is greatly considered as disease which Th2 cells predominate, our results indicate that the frequencies of Th1, Th17 cells are increased in lupus patients with or without renal involvement compared to healthy control, and that the Th balance in lupus patients is not shifted to Th2. So this suggests that the Th1, Th17 differentiation mechanism are altered in lupus, and the imbalance, the Th1, Th17 imbalance might be responsible for an increased pro-inflammatory response particularly in lupus with renal involvement. But also other and further research is needed to explore the underlying mechanism and also to validate this finding. So at the end, I would like to thank all members and researchers of the autoimmunity, cancer, and immunogenetics group and hospital in Tunisia. On behalf of all the co-authors, I would like to thank the BSA for giving me this opportunity to share my work with the community. And I want to thank the patient and healthy control also who participate in this study. And special thanks to the ICGAB, the International Center for Genetic Engineering and Biotechnology for funding this mobility. And that's all. Thank you.